So with plant invasion, it's very obvious what's going on above ground, but we know considerably less what's going on below ground. And that's where my interest lies. And because it's so obvious looking at the landscape here at MPG Ranch that there are areas that have been invaded completely, but we don't know what's going on in terms of nutrient processes and changes in microbial communities below ground, and that could have direct effects on the invaded community and also for our attempts to reestablish native communities. So that's really what the bulk of my project is about. And the first year we're just trying to get some baseline data and measuring differences between invaded communities and native communities. And what we're focusing on are ecosystem processes. And especially we're measuring decomposition, and in order to do that we're putting these litter bags on from different plant materials to see how quickly they decompose. And we're measuring potential shifts in nutrient dynamics by burying these resin capsules. And because decomposition is very dependent on moisture and temperature, we're also following changes in moisture, and by just measuring soil water content over the season and temperature. And to do that, we buried these small temperature probes. As sort of an end product, when organic matter is decomposed, it also emits CO2. So we're measuring soil respiration. And we expect that to be different in these different areas, native and invaded areas, simply because biomass production is so different and the timing of biomass production is so different. So let's go and take a look at the different plots we have and what we're doing in each plot. And we can start here with the native plot that is just nearby. Okay, so this is the native plot, and this will be our control in everything we measure. So this is the idea here, being that this is how these processes were before they were being invaded. So these are the litter bags, and what we did was to collect dead standing material from last year, essentially what will decompose this year or in subsequent years, from all 4 different plants that we're working with. So we have knapweed and leafy spurge, cheatgrass, and a mixture of native grasses. And this is a very straightforward way of measuring decomposition. We essentially know the weight when we put them out, and then after 3 and 6 months we will measure how much they weigh. So the weight loss is essentially what has been decomposed during that time. And we're using this reciprocal transplanting approach because we want to see how quickly leafy spurge decompose compared to native grass, but we're also interested in different decomposer communities. For example, the decomposer community that we find here is that specifically acclimated or adapted to decompose native grasses? Or does it do equally well with leafy spurge? And then compare decomposer communities in different areas. And this is particularly interesting for leafy spurge because it contains this white latex. So you could imagine that it would take some specific bug to actually decompose that. Okay, so this is the same approach here, but we buried it with roots. So instead of dead shoot material, we have roots, because we want to look at both components, and it makes sense to bury the roots because that's where the decomposer community will encounter that material, whereas the shoot material we have on the surface. And this is how we've installed these permanent PVC pipes about 3 centimeters down into the surface, and this is how we're going to measure CO2 emission, essentially soil respiration. We're just putting a light-core on here to measure how much CO2 is produced from here. And this will allow us to both look at seasonal changes in CO2 emissions as well as differences between invaded and native plots. So this is what we're using for moisture. We're essentially buried PVC pipe to a depth of 10 inches and 5 inches. And we have a probe where we can go out over the season and measure what is the moisture at 10 inches and 5 inches and then at the surface. Okay, so we're going to measure soil respiration, and this is what we use to do that is a light core. And essentially this measures CO2 flux. So what it's programmed to do is measure the time it takes for CO2 to emit from the soil 10 ppm essentially. So we put that on, and this is the PVC pipe we looked at before. Put this on and we hit start. Essentially now it's collecting all the CO2 that is respiring up and just recording the time from when we have set the ambient value, and it records the time it takes from going 5 ppm below ambient up to 5 ppm above ambient. And obviously the more CO2 that is produced, the quicker that time is. This machine is programmed to do the same measurement 3 times, and then it's giving us an average for the time of those 3 times. Yeah, so now we're in a cheatgrass area, and I chose these areas about 6 weeks ago by just simply basing it on dead plant material from last year. So in this particular plot, there's a lot of dead cheatgrass from last year. And we see new cheatgrass growing, and this is one of the characteristics of cheatgrass, that it grows so quickly. It's ready to flower, and it's just early May. And that's why it's so successful with the natives, because it goes in and grows early, takes up that moisture, early season moisture, and when the natives get going, the cheatgrass has already used up the moisture. So that's one strategy that cheatgrass has. So we're standing in a leafy spurge plot here, and you have to ask yourself how processes differ from this area compared to the native area we're just at. And that's essentially what we're trying to figure out. Not just the fact that it looks so different above ground, but trying to figure out what the differences are below ground. And yeah, leafy spurge is a really interesting one because it has this latex, and it's actually can be toxic and an irritant. But goats love it, and they actually grow better from eating spurge. So it's used in some other restoration projects, having goats and sheep go in and graze it. Yeah, and as you can see in the leafy spurge plot, we have the same setup as we did in the native plot in terms of measuring moisture and decomposition and respiration. It's just fascinating to me how spotty the vegetation is, because now we're moving into knapweed, and it's just covering this whole hillside. Then we still have the native vegetation over there, and cheatgrass coming in a little bit where it can, and then patches of leafy spurge. And what causes this patchiness, we don't really know. If it's just the soil conditions are slightly different and favor different plants, or the seed happened to fall there and it just takes off and grows from there. We don't really know. So this is the last plot. This is our knapweed plot, and as you can see, we have the same things here with the litter bags and soil moisture tubes and the PVC pipe for the CO2 measurement. And this is a good plot. It's not good, obviously, to have 100% almost knapweed, but it's good for what we're doing. In terms of hypotheses, I would Obviously, biomass is going to be a lot lower in cheatgrass, and I would think over time that we will see lower soil organic matter content in the soil, probably lower CO2 emissions or soil respiration. In terms of nutrient dynamics, I think it will be offset in time with cheatgrass, because it dies, it grows now and it dies probably in early June, and so that means you have all this dead plant material decomposing in June as opposed to September and the other plots with native plants and knapweed and leafy spurge. So very different nutrient dynamics. It's harder to predict differences we'll see in native communities and knapweed and leafy spurge, I think, because they're perennials. But in terms of knapweed and leafy spurge, we essentially have them as monoculture, which means that when that plant dies, everything will happen at the same time. Whereas the native communities, they're more diverse, so some plants will die at a certain time or grow and take up water at a different time. So I think that the differences are going to be in when things happen, and that it will be less variation in the native plant communities. Whereas we may see nutrient flushes in the leafy spurge and knapweed that we don't see in the native communities. And that's incredibly important for ecosystem processes. I mean, how much nutrients are lost from the system, and when that happens, if there's nothing else growing at that time, for example, to take that up. It's interesting. Although, they grow quite long into the season, so I don't know if we necessarily will see nutrient losses in those. But I could definitely see it on cheatgrass if there's nothing else taking it up when it decomposes. And soil erosion, for example, with cheatgrass, if it dies in July, if you have a bare soil after that, if they don't start germinating until October, November, you could essentially have bare soil. And if you have heavy rain, you could have tremendous soil loss in those areas. So those are all abiotic response to nutrient dynamics, but they're obviously driven by soil biota. And that's another component that can be drastically different between these different areas. And that is something that we're going to focus on and try to not just talk about soil biota, but try to separate it into different components. Out here we're looking at decomposer communities, but I'm particularly interested in mycorrhizal fungal communities, for example, because we do think that they're tremendously important for plant growth, both fungal abundance and community composition. So that is something we're also monitoring over the season, and to see how both those parameters may differ with plant invasion and what the functional aspect of that can be. And as part of that, we're trying to isolate these fungal species in the greenhouse so we can do some manipulative studies with them. So this is essentially what we're doing in the field, and we have a few things going in the greenhouse as well, so we can go and take a look at that. Okay, so a big issue in restoration is trying to get native plants to reestablish after you've gotten rid of the invader, and that's often done by just herbicide application. And the assumption there is that you can simply go out and just spread some seeds of natives and have them establish and grow as they did prior to the invasion. But there's good reason to believe that many factors change as an effect as a result of invasion, and those changes could be both abiotic and biotic. Abiotic being changes in nutrient dynamics, and for example, there are some indications that soils actually become more fertile after knapweed and leafy spurge. They produce more biomass that then decompose, and that's part of what we're measuring in the field to look at changes in these ecosystem processes. But it could also be biotic changes in soil microbial communities. And in this experiment, we're trying to, first of all, see if there are differences in native reestablishment after in soils that have been invaded for some time by cheatgrass, leafy spurge, knapweed, compared to native soil that had never had any invaders growing in them. And trying to separate the abiotic component from the biotic component. So we've simply collected soil from the different invaded areas we're working in and planted a forb and a grass. And the forb here is just flax, and a grass, the grass is blue bunch wheatgrass, which are common components in these reestablishment mixtures. And we have two treatments. We have a control where we're just watering and seeing how plants grow, and then we have a nutrient addition treatment with the hope to see differences there or separating the abiotic from biotic factors. For example, if differences are still apparent between the different soils even after fertilization, there is a good indication that there is a biotic component causing these differences. So we're simply going to monitor the growth of these. These were planted only 2 weeks ago, so they're only just now germinating and getting going. So there are no obvious differences yet, but I would be surprised if we don't see that as time goes by. So in this first experiment, we're simply trying to separate abiotic from biotic components and the soil legacy of plant invasion. And the biotic component can be obviously many things. Bacterial communities can change, and soil fungal communities can change. And we have reason to believe that arbuscular mycorrhizal fungi could be an important component during plant invasion. And arbuscular mycorrhizal fungi colonize approximately 90% of all plants out there. So they are a fundamental component of an ecosystem, and they help plant acquire nutrients. And native plants differ in the dependency on mycorrhizal fungi, but in general, forbs, for example, lupins and whatnot, are highly dependent on mycorrhizal fungi. And as an invader comes in, it can do two things. It can change the abundance of mycorrhizal fungi in the soil, but also the community composition. And both those things can have impacts on the functioning of the community. And these invaders can have different impacts on both community composition and abundance. And the three invaders that we're focusing on, knapweed, leafy spurge, and cheatgrass, which we have all here, are likely to respond very differently and impact differently the fungal community. For example, leafy spurge and knapweed are coarse-rooted perennials, and we suspect them to be heavily dependent on mycorrhizal fungi and actually enhance possibly the abundance of fungi in the invaded soil, whereas cheatgrass is a fine-rooted annual and it could potentially decrease the abundance and change the community composition. And that could have an effect on native communities when we're trying to reestablish them. So that also ties into the first experiment I was talking about, because we could see those changes. For example, if we're trying to reestablish a native that is highly dependent on mycorrhizal fungi, and we have had cheatgrass there previously, it may do very poorly, and we might actually have to amend the soil or actually increase the mycorrhizal fungi for it to reestablish successfully. So in this portion, we've simply transplanted plants from the field, from the 12 different sites where I'm working. So we have cheatgrass, leafy spurge, knapweed, and native plants growing here, and with the idea that we can make arbuscular mycorrhizal fungi sporulate. So if we can make them sporulate, we can take spores and start single species cultures. And if we have that from these different sites, we can actually start running experiments and inoculate plants and see how important mycorrhizal fungi are for these invaders as well as the natives. So we're trying to get to a more finer scale, taking the biotic community or biotic component from the first community, and then actually isolating one component of that, the arbuscular mycorrhizal fungi, to allow us to manipulate those things. So that's what we have going here. Here's the same thing, essentially soil from these 12 different plots, but we're growing the same plant in all different soils. And this is just sorghum grass, which function as a good host. So it gives the fungus a lot of carbon, and we're hoping to get a lot of different spores from here to start single species cultures.